All right, here we go in 5, 4, 3, 2.
Hello, everyone. Welcome to this wonderful panel. If you are in the Interact and React poster session, that is where we are with our wonderful panelists. I'm one of the moderators today. My name is Annie Zeidman-Karpinski. I am she, her, hers. And we also have
Hi, I'm Andrew Wang. I'm the other moderator doing mostly back-end stuff. My pronouns are he, him, they, them, and I'm excited to get this started.
So I was looking at the Trello board and I thought we'd go in the order of the Trello board, and then the first one would be Jasmine Cole, but I'm not sure that she's here, that they're here. Is Jasmine here? My apologies. Okay, the next one, Alena. Alena is here. Are you ready to speak?
Yes, I am. Hello. And I can actually just share my screen since I have my poster up. Okay, let's just get it. Just try to finick with it so that it's full screen. There we go. Does that look okay? You can see the whole poster. Awesome. Hi, everyone. So my name is Alena Silicon, and I'm an undergraduate researcher in Dr. Diana Labuda's lab. And my research pertains to defining the roles of conserved DNA repair complexes in the maintenance of C. elegans meiotic genome integrity. And really, I'm just, the main focus of my research is to study DNA repair in meiosis, which is a specialized form of cell division that many sexually reproducing organisms utilize to create sperm and eggs. And inaccurate DNA repair that can happen during meiosis can lead to detrimental errors, as well as the development of various diseases such as infertility, cancer, and birth defects. And so the main focus of my research is to study 3 conserved DNA repair complexes and their interactions in promoting DNA repair. And those are shown here on my poster. It's the structural maintenance of chromosomes 5-6 complex, NSC2, which is a subunit of this complex, and the BRCA1 BARD1 heterodimer that's commonly mutated in mammalian breast cancer. And I use a variety of cytological approaches and as well as genetic approaches in order to answer my question. And really, the biggest findings from my research are that although NSC2 and SMC5-6 have been studied in other organisms, no work has been done on NSC2 previously in C. elegans, and I implicate a role for NSC2 in a response to DNA repair and meiosis, essentially. And then additionally, I also, no previous research has shown how these protein complexes interact genetically to promote DNA repair. And I find that they're differentially regulated in meiotic prophase 1 in order to resolve DNA repair. So that in sort of the 2 broad windows of DNA repair, which are prophase 1 of meiosis, there's early to mid prophase, as well as late prophase. And in these different windows, different DNA repair pathways can be engaged. So in early to mid prophase, DNA repair is regulated through this linear pathway. And in late prophase, we see that it's regulated through a diverging interaction pathway between these proteins. And a really interesting finding is that we hypothesize that there may also be some suppression of potentially mutagenic repair pathways that's also occurring in late prophase 1, and that these proteins are functioning in. So that's kind of broadly my research, and I look forward to hearing any questions or feedback.
Do we have any questions from either other panelists or from folks who are attending? And you're welcome to unmute yourself and ask the question, or if you want to put it in the chat, we are ready to read your question out loud to everyone. And we can do Q&A at the end. I just want to give folks a chance who might be having to drop in and out to ask some questions while they're still fresh.
Okay, yeah, so there's a question about why anti-mutation pathways may be favorable in prophase, and that's a wonderful question. And so So meiotic cells can utilize 3 different repair pathways broadly. There are 3 different types of repair pathways. There's interhomolog crossovers, which are also known as recombination, and this results in essentially error-free repair. And then they can use alternative DNA repair pathways, which are broadly characterized as accurate and error-prone. And it is known that meiotic cells prefer to use accurate repair pathways. And the reason for this is that those error-prone pathways can result in mutations when they try to repair DNA. And what's really sort of the crucial aspect of suppressing those pathways is that any mutation that we acquire, even if we're fixing DNA, any mutation that we acquire in sex cells will then be further passed on at fertilization, so that it essentially is, if we imagine sort of a sex cell, that sex cell is going to fuse with another sex cell, and then that one cell now is going to divide, and it's going to form every single cell in the progeny's body. So any mutation that would occur in a sex cell, we would see it in every single cell of the body after fertilization and development. And so that's usually why those pathways are suppressed so heavily.
Does anyone else have questions at this time for Alena, who did a marvelous job of presenting this amazing work? Okay, we're going to go on to Lisa.
Yes, okay. So I'm going to go ahead and share the poster. This is actually a joint presentation with my lab partner, Noah, so we'll be doing this together. Right. All right. And Noah, do you want to start off?
Yeah, absolutely. So yeah, my name is Noah. My lab partner, Lisa, and I have been studying the impacts of environment on the degree of despotism in semi-free-ranging Japanese macaques. So our research centers on the social hierarchical system in the Japanese macaque, which is a social primate species that is native to mainland Japan and some of its surrounding islands. Primate societies have evolved social hierarchies where higher ranking individuals gain better access to resources such as food and mating opportunities. Different primate groups have different methods of maintaining these social hierarchies. So we categorize their level of hierarchical flexibility on a spectrum from tolerant to despotic hierarchies. So tolerant leaning hierarchies are typically seen in environments where cooperation is important for individual success. So these are characterized by moderately high rates of low intensity aggression, low rates of severe wounding, that is, and a less linear dominance hierarchy. They also show higher rates of positive interactions within close proximity to one another, such as huddling and grooming. So in contrast, despotic leaning hierarchies are seen in environments where individuals gain benefits by competing against each other for access to resources rather than cooperating. They're distinguished by high rates of wounding between individuals and a highly linear dominance hierarchy with infrequent interactions between individuals of different ranks. So although Japanese macaques groups are generally described as despotic leaning, primatologists Zhang and Watanabe have found that there are actually some groups which demonstrate a more tolerant leaning hierarchy. So socio-ecological or environment factors such as provisioning, sex ratio, and genetic isolation also may influence this intra-species variation that we've seen in dominant style. So in order to assess our study group's degree of tolerance, we compared our study population to the provisioned groups from Zhang and Watanabe's article.
Yes, and our study group is comprised of 17 adult male Japanese macaques at the Oregon National Primate Research Center in Beaverton, Oregon. And these males were between 7 to 25 years of age and identifiable by dye markings on their backs. Like Noah was saying, we followed the methodology of the Zhang and Watanabe paper and calculated the mean rate of aggression per hour per individual, along with the standard deviation. And this was using the 512.5 hours of behavioral data collected by our team to look at instances of, as well as frequency of, aggressive behaviors. And this allowed us to conduct a preliminary comparison of our study population against the 3 populations studied by Zhang and Watanabe. After we analyzed the ONPRC data, we found that out of the 32,800 focals inputted, 115 were instances of aggression, which meant that, this meant that the ONPRC group that we're observing demonstrated a mean rate of 0.3 aggressive interactions per hour per individual with a standard deviation of 0.2. And this is most directly comparable to the Shotoshima Island population, which also had a mean rate of aggression of 0.3 aggressive interactions per hour per individual with the same standard deviation.
Yeah, so these preliminary results did show a promising comparison between our study group at the ONPRC and the tolerant leaning Shotoshima group, potentially inferring that our group is also tolerant leaning. However, further comparisons between these 2 populations are definitely necessary to determine our study group's degree of tolerance. So we also plan to measure the intensity and frequency of wounding, the percentage of active time grooming between adult individuals, and frequency of positive interactions. So some theorize that these intraspecies differences in dominant style that we've observed can be attributed to the varying degrees of nepotism in each group. So more despotic leaning groups will promote higher nepotism and kin support, whereas more tolerant leaning groups actually promote higher non-kin support. And both the ONPRC and the Shotoshima Island groups have been genetically isolated for multiple generations, as Shotoshima Island group has been isolated on a fairly small island for several generations, while the ONPRC is in a semi-free ranging corral. So this would result in higher degrees of interrelatedness, which could explain their more tolerant leaning behaviors. So the high variation in dominant style seen in this Japanese macaque group indicates that it's potential that dominant styles are environment specific rather than species specific, which is why we do see this intraspecies variation. And this research that we've been doing supports previous evolutionary research into the conditions which favor the evolution of social bonds. Yeah, so we welcome any questions or feedback.
So here's a question: what other behaviors might not be species specific, but a response to different environments?
Yeah, that is a great question. So dominance hierarchies are actually an example all across the board of where we see a variation within the same species, but in different groups. And dominance hierarchies essentially dictate the social relationships between all of the individuals in the group. So behaviors that we would see that would essentially be a response to different environments include the amount of time individuals spend grooming one another, and also just the amount of time they actually spend within 2 meters of one another, or the comfort they feel approaching one another, and in general, just cooperation in terms of resource allocation and general social behaviors. We actually see at times a difference within a species, but in different groups.
Yeah, and that's why we're so interested in the Zhang and Watanabe paper, because while Japanese macaques are typically despotic, they observed this one very tolerant group, and we found, we wanted to test if we could see, like, statistically, if there were characteristics of this tolerance behavior in our own PRC group here in Beaverton.
Right, because the environment in terms of like the climate and the density of trees, et cetera, resource allocation, all of that was very similar among all groups. The main differences between these tolerant leaning groups versus these despotic leaning groups was mostly genetic isolation. Any Japanese macaque group that's been studied so far that has been on an island has been shown to be tolerant leaning. And so it would make sense that our group, which is in a semi-free ranging facility at the ONPRC, that already have very high degrees of interrelatedness, would show these same tendencies.
Thank you both so much for this exciting work. What are you hoping to do next?
Yeah, so we were hoping to look more at, so right now we have data on frequency of aggressive behaviors, but because a lot of the 3 other populations that in the Jung Watanabe paper also had like similar levels, I would say, of aggression. There are so many other factors that need to be analyzed to see if the population at the ONPRC is tolerant. So we wanted to look at rate of grooming, rate of frequency of approaches, and some of the other behaviors that Noah was talking about that leaned to tolerance.
Right, and we also have the privilege of having access to some of the health records of the primates at the ONPRC. So that'll allow us to look at frequency of severe aggression, which is a characteristic of despotic groups. So we're hoping to gain access to those health records and see how often these aggressive behaviors lead to more high intensity aggressive behavior, which will allow us to determine the degree of tolerance of our group.
Thank you both so much.
Because oh wait, can I say one more thing? Sorry.
Yes, please.
Yes, we observed that in a lot of tolerant groups, aggressive encounters might actually be more frequent, but they'll be less severe. So like Noah was saying, we would check the records to see the intensity of these aggressive encounters and like kind of like the severity of the wounding, and that would help us categorize them as tolerant or despotic.
Thank you both.
Thank you.
Thank you.
We need some sort of way to like signal like that you're sitting down. So thank you so much. We're going to move on to the next presenter, and that is Abbey.
Hello, everyone. Let me share my screen here. Okay. So hello, everybody. My name is Abbey Ward, and I'm part of the Early Dual Language Development Lab in the College of Education. And today I'll be talking a little bit about my honors thesis, which is on lexical semantic processing in bilingual toddlers and how language proficiency is influencing this. So there's recent research being conducted in the field of psycholinguistics that suggests lexical semantic networks, which we refer to as vocabulary systems, interact with one another in bilingual individuals. So my first research question was aimed at answering whether or not vocabulary systems interact across languages in bilingual children as young as 2 years of age. So we wanted to know that, is it that the words perro in Spanish and cat and their concepts overlap, or are these two words relatively separated into two vocabulary systems? So this is demonstrated here in figure 1 of my poster. And then secondly, we wanted to know that if these vocabulary systems interact, then what are the sources of variability that predict this organization? So my second research question looked at whether proficiency, which are the words that a child says and understands, versus exposure, or the words that a child hears in each language, influence this hypothesized cross-language interaction as shown here in figure 2. So to measure both the cross-language interaction and also proficiency and exposure, we'll move to our method. So there were 20 toddlers that with approximately 2.5 years of age with dual language exposure to Spanish and English who participated. And as a group, they were exposed to Spanish more than English. We included toddlers with evidence of early language delay in order to assess language proficiency across a continuum. And then we had 4 measures to answer our research questions. So to answer the first question on this cross-language interaction, we used an eye tracking system to measure toddlers' processing of semantically related and unrelated words in 4 different language conditions within and across Spanish and English. So for example, as shown here in figure 3, a toddler was presented with the prime word jacket, and then they heard the related target coat. Then they were presented with a pair of distractor and target images. And then during this image presentation was when we assessed their looking behavior and calculated the prime effect, which is the proportion of total looks in related trials minus unrelated trials. Then moving to our independent variables of interest, the language exposure assessment tool is a parent report measure which captured these children's exposure in both Spanish and English. Then we have the English-Spanish vocabulary inventories. This is also a parent report, and it measures expressive vocabulary size, which are the words that a child says. And then we have the computerized comprehension task, and this is where children are prompted to touch a target image, as seen here in figure 4. This was a measure of children's receptive vocabulary, so the words that they understand, and then also their speed of word recognition. So moving to our results, what did we find? So remember, my first question was interested in knowing whether vocabulary systems interact across languages. In figure 5 here, we have children's proportion of total looks to the target object on the y-axis, and then on the x-axis, we have 4 different language conditions, which looked at within and across language processing. So the dark gray bars are the related trials, such as jacket and coat, and then the light gray are unrelated trials, for example, jacket and dog. Then the line at 50% indicates that children look to the target above chance levels. So our results indicate that when the prime word was in Spanish, children had a greater proportion of looks in the related trials than the unrelated trials. And then when the prime word was in English, we see this reverse effect where toddlers had a greater proportion of looks in the unrelated trials than the related trials. And then we can see that this is significantly different in the within English block. So when an English word primed an English target. Now moving to my second research question on proficiency versus exposure. Here we have our Spanish priming results in table 1. So when the prime word was in Spanish. I've listed the correlations between the prime effect and our measures of proficiency and exposure, and the significant effects are bolded. So we first note that language exposure was not correlated with the prime effect in both Spanish language conditions. And then moving to proficiency, we found that this was a better predictor of lexical semantic processing. Overall, you can see that children with large receptive vocabularies and slow reaction times had the largest prime effects in Spanish. And then moving to table 2, this again shows correlations between the prime effect and measures of proficiency and exposure, but this time when English was the prime word. So again, we see that relative language exposure was not correlated with the prime effect. And then in English, we see that a faster reaction time correlated with a larger priming effect only in the within English condition. So what can we interpret from these findings? Well, from our results, we see there's evidence for cross-language interaction in young dual language learners 2 years of age, but these numerical differences are not significant. But then what explains the different pattern of findings in English versus Spanish? So our results suggest that young bilingual children are sensitive to semantic relatedness in their stronger language. And then when we are in their weaker language, in English, children use these sparse vocabularies in such a way that knowing fewer words is leveraged to support correct target identification. Now, we answered a second research question by finding that proficiency rather than exposure was the better predictor of lexical semantic processing, both within and across language conditions. So as a group, toddlers with larger vocabulary sizes and faster speed of word recognition in their stronger language or Spanish demonstrated inhibition such that they were more likely to discard words similar in meaning in order to correctly identify the target. And this is represented here by figure 8, where multiple competing links in a network result in inhibition. Now, in processing words, word relatedness between words in their weaker language or English, toddlers with slower speed of word recognition demonstrated facilitation. And this may offset the fact that they have a sparse vocabulary, demonstrated in figure 9. Oh, I have. So then in figure 10 represents our findings that language exposure was not associated with lexical semantic processing. And rather, we conclude that proficiency measured by vocabulary size and then speed of word recognition was a stronger predictor of lexical semantic processing. Thank you all. Any questions or feedback?
We have one question from the chat. It says, do you expect the cross-language interaction to change as bilingual children get older?
Cross-language. That is a great question. And off of the top of my head, I'm not sure. My guess is that these children that participated in our study, we categorize them as simultaneous bilinguals, so children who are learning 2 languages simultaneously. For children who are maybe they establish their first language, and then later on as they get older, they enter school, those would be considered sequential bilingual children. And so there's probably going to be a difference between those 2, sequential versus simultaneous. My guess is that as they get older in these simultaneous cases, that children, they'll have stronger links between their networks and they'll be able to kind of maybe code switch between them. But that is a great question that I think would be really interesting to look at. And I'm personally not aware of literature on that, but I will ask my mentor because I'm sure there is.
And then a follow-up question, why did you choose toddlers?
So the kids who were in our study were about 2 to 3 years of age, and the reason that, oh yeah, kind of, but one of the reasons, the other reason that we chose toddlers around this age is that this is when their vocabulary systems are emerging. They're beginning to put together multiple sounds and make words, and they're also beginning to have utterances. So they're starting to string together 2 to 3 words in each of their languages.
Thank you. My last question for you, what inspired you to do this work? Like, what was the biggest inspiration?
Well, I joined the EDLD lab the end of my sophomore year because I wanted to get involved in the lab so that I could kind of explore a thesis topic. And then I started talking with my mentor, Dr. Stephanie Deanna, in the lab, and she was like, you could use the eye tracker, which is really cool. And if you ever have a chance, please look up eye tracking, like in the psychology, because it's really fascinating. But I kind of wanted to look at bilingual language development broadly, but that's a very broad topic. And so she kind of helped me narrow down what exactly we could look at. And because we have in our lab, we have so many different measures that we can use to look at what children are hearing in their home environment. And then we can bring them into the lab and see how they're developing their languages and what words they're knowing. So just bringing together all those resources, I think, was a really cool opportunity. So yeah, lots of inspiration, lots of things that I'm still thinking about working on later on down the road.
Fantastic. Thank you so much.
Yeah.
Thanks for all your hard work on this. Next up is going to be Byron Lee.
Hello, everyone. Can you see my screen? Okay. All right. Let's see. Okay. So, hi, my name is Byron. Today we're going to be talking about ROBO4 and its relationship to Alzheimer's disease. So, Alzheimer's disease has been discovered for about 118 years, and it was discovered by a scientist named Aloysius Alzheimer back in 1901. But essentially, there have been no significant progress in treating Alzheimer's disease since 1901, and this disease is becoming more prevalent worldwide. According to a 2019 Alzheimer's report, it is predicted that the number of Alzheimer's disease cases will double by 2050. And so in the Aging and Vascular Physiology Lab, we believe that understanding the vascular system is key to creating potential treatments for Alzheimer's disease. And so in your brain, you have a system of blood vessels, and the blood-brain barrier basically divides your blood from the extracellular environment in which the neurons reside in. Now, we don't want any neurotoxic compounds to enter our brain tissue. And so therefore, this barrier, it's a very restrictive barrier, meaning that it only allows very few substances to pass through. But as you age, your blood-brain barrier may not be that restrictive barrier as it once was. So now this is a zoom in on figure 1. So figure 1, you could potentially see why this could be a problem. So you have neurotoxic compounds, you have red blood cells that can make its way into the brain tissue, and this can cause inflammation. And inflammation can decrease amyloid beta clearance. Now this could potentially lead to an increase in amyloid beta plaque formation, which is a common characteristic seen in Alzheimer's disease patients. So the question is, what can we do to prevent increased permeability? Well, we believe that this ROBO4 protein receptor can inhibit something called vascular endothelial growth factor, or VEGF, which inhibits angiogenesis, or the formation of new blood vessels. So we don't want to form new blood vessels in this case, because in order to produce new blood vessels, we actually have to increase the permeability of those existing blood vessels, which is not what we want in this case. And so we conducted a study where we used ROBO4 wild type, so that's the 2 pluses over there, knockout, the 2 minuses. So a knockout means that this protein receptor is not present in mice. And we did that with APP mutation and without APP mutation. So APP stands for amyloid precursor protein, which leads to the overproduction of amyloid beta. Now, this is important because in mice, they don't normally get these amyloid beta plaques formed in their brain. And so therefore, we have to genetically introduce that through APP mutation. And so during the experiment, we hypothesized that old mice will exhibit greater cognitive dysfunction than young mice, and ROBO4 knockout mice crossed with APP mutation will exhibit greater cognitive decline. And to test this, we use Moore's Marmaze and nest building tests. So right here, we have our nest building test, and each mice was placed into separate cages with a paper nestlet, and they were left overnight. And so we rated each nest between 0 to 5. So if the score was 0, no nest was formed. But if the score was 5, a perfect nest was formed. So what I mean by perfect nest is that the nest is circular, it contains a crater, and is higher than the mice's body. Now going to the water maze, so we conducted this test for 5 days. So on your left, you have a tub of water, and in that tub, there's a transparent platform that is submerged underwater. So our goal here was to allow the mice to determine where the platform was. Now, if you look at the figure on the right, you see that there are numbers around this tub, and those are drop zones. So we would basically drop a mice into different drop zones for different trials, and we hope that they would learn where the platform is. And so for day 3 and 5 of the experiment, we conducted the probe trial, meaning that the original platform was removed. And so from this picture on the right again, the probe trial measures how long the mice would swim in the target quadrant of where the original platform was located in. Now going to figure 2, so we can see this is the Moore's water maze result. So we see at day 3, there isn't a significant difference between the young and old mice. But if you look at day 5, or the last day of the experiment for part B, there was a significant difference in that the young mice spent a great amount of time in the target zone. And this indicates that they learned where the platform was better than the old mice. Now, figure 3 here shows the results for the nest building test. So for part A, we can see that the young mice had significantly higher nesting scores than the old mice. And so for part B, we looked at ROBO4 wild type, knockout, crossed with APP mutation and without APP mutation. And we can see that there isn't a significant difference between those 2 groups. So we can conclude from our results that old mice had impaired cognitive function when compared to the young mice, but there wasn't any significant difference between ROBO4 knockout or amyloid beta overproduction. So we hope to conduct future tests by comparing ROBO4 with APP mutation for cognitive function to measure blood-brain barrier permeability in ROBO4 mice, in addition to examine amyloid beta and neuroinflammation in brains of ROBO4 mice. And so although this set of studies did not find a role for ROBO4 in cognitive impairment, we need to perform further studies before making definite conclusions. So our continued studies in this area will help us better understand the role of the blood-brain barrier permeability and the ROBO4 protein receptor in Alzheimer's disease. Thanks for listening, everyone. Do we have any specific questions? Okay, I'm gonna start sharing my screen.
Great job, Byron.
Thank you.
We have one now. What other cognitive tests would you like to explore?
Cognitive tests that we would like to explore. Well, there are many types of cognitive tests we can conduct. We did the water maze, we did the nest building test. I believe that there's another test where I think it would be pretty interesting. So like we read an article and it's basically like sort of a maze. So they would actually have to find, for example, like a specific object in that maze. So I think it would be pretty interesting to investigate, like compared to the water maze and the nest building, like would the mice be able to find where that a particular object is? And I think that's pretty interesting. Yeah, but there are a lot of cognitive tests out there for sure. Okay, we got another question. So the question is, are there any direct ways you can measure the permeability of the blood-brain barrier, like introducing some impurity into the bloodstream and measuring how much of that impurity ends up in the brain? That's a very good question. And actually that is the next step to our research. So basically we're gonna do something called retroorbital injection. So basically we would inject the dye into their eye, and then that dye would flow around their circulatory system. And eventually that would allow us to determine whether there was any leakage of that blood-brain barrier. And so that is one of the next steps. Very good question. Do we have any more questions?
I have one last one. So what's something that surprised you?
What surprised me was just in Alzheimer's disease in general, there's a lot of things we don't know about, like ROBO4, blood-brain barrier permeability. That's just one mechanism of Alzheimer's disease. There's plenty of mechanisms that could potentially cause Alzheimer's disease. So that's why Alzheimer's disease, it's a very hard disease to tackle upon, because there's just so many factors and there's just so much we don't know about. So when I was actually doing this project, I was kind of, I was really like surprised to know, like, wow, there's so many other factors that could be affected. It's not just like this protein or this blood-brain barrier permeability. There are other factors.
Fantastic. Thanks for that wonderful presentation and for all of the wonderful questions that we got from the audience.
Right, thank
Okay,
you.
thank you so much. Next up is Ian.
Yeah, I believe now you can see my screen.
Ian, can you turn up the volume at all?
Or I can talk louder.
Thank you so much for doing that.
Yeah, and that might also help a little bit. Okay. Hello everyone. I don't know if that, do you guys still see my screen? No, I'll just leave it like this. Hello everyone. My name is Ian Torrence. I work in the DWJ lab with Darren Johnson as the PI, and I work to develop little chemical sensors to detect various different ions floating around in solution. And what I'm going to talk to you today about is one of those chemical sensors to detect ammonium, the ammonium ion. And the reason why I wanted to focus on the ammonium ion for this project is because today, sort of in the Western world, farmers tend to over-fertilize their crops using nitrogen-based fertilizers. And one of the main ingredients in those fertilizers is ammonium nitrate. And so this is somewhat of a problem, because when you over-fertilize the crops like this, when it rains, a lot of the nitrogen through ammonium and then also through the nitrate ends up in sort of our water bodies, so lakes, streams, rivers, etc. And this induces eutrophication or massive algae bloom growth, which sucks a lot of the oxygen out of the water and kills the local fish and also disturbs local wildlife. And so I developed essentially a chemical field effect transistor, or a ChemFET, to selectively sort of detect ammonium in some sort of aqueous water environment. And the main way we do this is essentially by setting up test tubes, essentially with various different amounts of concentration of ammonium, and then just kind of dipping the FET in them, waiting 5 minutes for the FETs to equilibrate, and then we get a response. The results of that type of test is seen here, where you have a linear portion of the graph, which corresponds to the FET sensitivity. In this case, the sensitivity is approximately negative 49.1 millivolts per decade, decade just meaning per log unit of concentration. A perfect sensitivity is about 60 millivolts per decade, or negative 60 millivolts per decade, which is given by the Nernst equation, although you almost never get sensitivity that high. Additionally, you also have a sort of flatline portion of the graph here, which corresponds to where the FET can no longer tell the difference between sort of the various background noise going on in the solution, or just of the FET itself, and then also it can't tell the difference between background noise and then any difference between sort of the concentration of your target ion, in this case, ammonium. However, these types of tests, while they are important to sort of get a general sense of how they do with your target ion, in the real world, there isn't just one ion floating around in your water system. You have a whole bunch of different ions kind of floating around. So you need to have some sort of test that sort of can tell whether or not your sensor can sort of selectively bind to and respond to just your target ion and not any of this other random stuff. And we do that with fixed interference tests, in which we still sort of steadily increase or decrease the amount of your target ion, or ammonium, but then you have also in the solutions a fixed amount of interferent across all the solutions. And these sort of, I've picked sort of 3 fixed interferent results down here for potassium, sodium, and calcium. And these results, the main sort of figure of merit that we want to look at here is the selectivity coefficient, which is essentially a measurement of the sort of responsiveness. So for example, if you get a selectivity coefficient, sodium is actually a really good example, of about 0.01, that means that the sensor is 100 times more responsive to ammonium in this case than it is to the sodium. For potassium, it's about 10 times-ish. It's not quite 10 times as much. However, for calcium here, the sensors are really, really selective to ammonium over calcium, which is good. So sort of the general sort of takeaways from these is that the FETs show good sensitivity to ammonium, even in the presence. As you can see, the linear portion of the graph are still pretty good. The slopes, while I don't have them listed here, all sort of fall within sort of, or all sort of agree with a sort of negative 49.1 millivolts per decade figure. And then also the fixed interference results all show that a pretty good selectivity for ammonium versus other ions. Some things I would like to do more on this project is adapt my FETs to actually look at soil samples rather than just look at water samples. Soil samples are a lot better because then we could actually like go in field to farmers' lands and actually take a measurement of their soil and see, okay, how much ammonium is in the soil. And one way that we can do this is using a fixed flow injection analysis, or FIA, which I hope to be able to present at the next year's symposium. Thank you, everybody, for your time, and I will take any questions.
Great. We have one question. Is it possible to get ChemFed out commercially so we can all test our own water?
Yes, there are some companies that already sort of work with this type of technology. Supersensor is one of them. It's not commercially for like everyday sort of general use. While you could do, this is more sort of directed towards industrial use. But you could, yes.
Marvelous. We're going to move on to Dylan next. And if there are additional questions, feel free to put them in the chat and we'll get to them at the very end. I just want to make sure everyone has a chance to present. So Dylan, you're on.
Hi. Oh, there we go. Can you hear me? Okay, I think so. All right, I'm going to try sharing my screen. All right. Let's go to... So, hi everyone. My name is Dylan, and I'm going to talk to you about iron and selenium. Before I do that, I'm going to preface by saying, with all of our increased time indoors lately, many of you may have found out that in your baking experiences, as we know, we're all making banana bread, that if you cook something too long, you get black, you get carbon. But if you cook it just the right amount, you can get a variety of different things. And this is the fundamental advantage of organic chemistry, is that if you introduce too much temperature, you'll get one thing. It's CO2 and maybe just carbon. But up until that point, you can get this broad variety of different molecules and compounds. Now, what I'm doing with iron and selenium is effectively that, except for now I'm working with inorganic elements. Now, the fundamentals are the same, and I'll introduce some terms here by saying that the CO2, the carbon that you get when you really burn something, well, that's the thermodynamic product. You give the material all the energy it wants, it'll eventually make this product. Now, what I'm trying to do in my research is to react iron and selenium in such a way that I don't get the thermodynamic product. Unfortunately, for inorganic compounds, and I'm actually working with a solid state material, so not a molecule, but more of a repeating array of atoms. Unfortunately, with inorganics and solid state chemistry, you get the thermodynamic product pretty quickly, and you don't have quite the diversity of intermediates you can get along the way before you get to the thermodynamics state. Now, in my case, why I'm interested in iron and selenium is because, well, a few years ago, people discovered that iron and selenium, when you mix them together in the right phase, it can superconduct, and it can superconduct pretty well at a pretty high temperature. And ever since then, everyone's been trying to make iron selenide, is what we call it, beta iron selenide, a particular phase of iron selenide. Everyone's been trying to make this, the structure, but we still don't really know exactly how iron and selenium react to make the structure and how we can best make it, get it in its purest state. So now this iron selenium, this beta iron selenide, it may or may not be one of those kinetic, we call it a kinetic state, where you haven't gone all the way to burning the material. You're kind of cradling it nicely in some not quite burnt state. So in order to do that, you've got to give it a little bit of temperature, but not too much. If you give it too much temperature, you'll end up with the thermodynamic product, which may or may not be iron selenide. I'll say that in beta iron selenide, this one that we're interested in, is in often cases a thermodynamic product, but I want to see how stable is it and how likely are you to form other things instead. And now in order to do that, we have to, in order to test the kinetics of the formation of irons and selenium compounds, we want to eliminate the problem of getting iron atoms and selenium atoms that are separated and get them to mix together to make this solid state compound. Because as you can see in a lot of these structures, you have a nice even dispersion of iron and selenium. You can't just take a block of iron and mix it with a block of selenium, because then you're going to have to actually heat it up a lot to get those two materials to mix. Now, I, in my research, can probe the kinetics of the interactions between iron and selenium by doing most of the mixing myself. And I do this by actually stacking them in layers, atom by atom. Now I say atom by atom, we don't have quite that much resolution, but we can do layers of atoms, layers of iron, layers of selenium, iron, selenium. So we don't have to do quite so much mixing, and we can run our reactions at a lower temperature and see what we find. Now we create these layered materials, these artificial layers of iron and selenium, by essentially vaporizing like pure iron and pure selenium. Now you may all know that iron is a metal, and in order to vaporize it, you've got to get it pretty hot. But once you do that, it'll actually it'll just start sticking to everything around it that's cold. It's essentially a beam of iron moving throughout what we call our deposition chamber. And eventually these iron atoms or these selenium atoms will stick to our substrate, which is just a silicon wafer that's kind of spinning around pretty far away from, or a short distance away from the source. Then, as you can see in this in my method section, you'll get your nice artificial layering of the two materials, and then we will heat it up a little bit or a lot, depending on how much we want to. Heat up our samples just by putting it on a hot plate or like a stove. And by giving it just a little bit of energy, we'll let those atoms relax and kind of form the structure they want. Or at least the structures they can access with the amount of energy they have. And then we'll take these samples that we've gently heated, and we'll subject them to X-ray diffraction and X-ray fluorescence to try to see how exactly these atoms are arranging themselves and if they formed the precise structure that we're after. Now, I'll save you all of the science behind X-ray diffraction and fluorescence, except for I'll tell you that every structure of iron and selenium that forms, and I'll tell you there's like 7 or 8 of them that are known already, they're going to have their own characteristic X-ray diffraction patterns. And that's what we see down here in the results section, where I've actually plotted these X-ray diffraction patterns as a function of temperature, which kind of increases as we go up. Now, to quickly summarize these results, I'll say that, I guess I should preface by saying I did this with 2 kinds of materials, 2 kinds of samples. Samples that have 1 selenium atom for every iron atom, or about 1 to 1, and samples that had about 2 selenium atoms to every iron atom. So I was looking at, mixing, just like you're making a cake or something, like what's the right ratio to get me a nice cake, or in this case, the beta iron selenide structure that I'm after. Now, in the 1-to-1 ratio case, it looks like we do form this beta iron selenide, which makes sense. It's 1 iron, 1 selenium atom, so it makes sense that we would form iron selenide. Now, there are other iron selenide structures that form, and I found that actually as you increase the temperature too much, to about 450 degrees Celsius when you're heating your sample, you'll form this other, we call it delta iron selenide, which isn't the superconducting structure that we are after. And then going over to the 1-to-2 ratio of iron to selenium atoms, looks like we don't form this with this iron selenide structure, which again kind of makes sense. Now we have twice as much selenium as iron, so a 1-to-1 product wouldn't form so readily. And actually, in the 1-to-2 ratio case, we get a diversity of different structures that form. We have an FeSe2, an Fe3Se4, and eventually an FeSe. We do go back to an FeSe structure because it turns out at really high temperatures, just like if you burn your bread too long, you'll get CO2 and carbon. If you burn iron and selenide or iron and selenium too long, you'll get this FeSe structure that we don't want. So really what I was doing in this experiment was seeing how easy is it to burn iron and selenium. And in this case, I actually found that it's quite easy. Now the thermodynamic state, whether it's beta iron selenide or delta iron selenide or FeSe2, that depends on the composition. So how much iron, selenium, and what temperature it is, or what temperature you've annealed it at. And it turns out that all of the structures I found that formed are pretty well described by thermodynamics, meaning that there aren't very many kinetic barriers to the formation of iron selenium products. Now, this could have kind of important implications for people who are trying to use iron and selenium to see if they can rearrange the atom slightly differently, such that we maybe even get a higher superconducting temperature, because that's really why people are looking at this material now. But On the other hand, I also found that this iron cyanide structure forms really easily. So we can probably make FeSe, beta FeSe, under some pretty easy conditions. Yeah. So I think with that being said, I will take any questions if people have them.
Dylan, we have one question already, and that is, so you're saying if you had an iron selenium baking pan, could you make banana bread in 5 seconds from room temperature?
You might. The danger is that iron also really likes oxygen. I think we're all familiar with rust. So when we do our baking, we have to bake it in an environment, in an oven that doesn't have oxygen in it. But yes, you could.
And the question was flaming banana bread. Thank you so much, Dylan. We're going to go to our last panelist, and we'll have some time to ask Dylan and Ian and Adeline, especially, questions at the very end. Okay, so Adeline, you're on. Thank you so much, Dylan, for that fascinating presentation.
Okay, hi everyone. I'm going to also share my screen. So my project was on the influences of sensory conditions on social behavior and brain activity of zebrafish, which is essentially, we're asking the question of what sensory input like vision, olfaction, or tactile senses is most important for social behavior, and how does that affect what parts of the brain are active. And so some background on this is we're interested in this subject, one, to understand social behavior, which is a part of everyday human life, but also understand times where there's social behavior deficits, like an autism spectrum disorder. And so zebrafish are actually really great models to study social behavior because they're highly social animals. And for us, we're looking at particularly 3 social behaviors. If you can see on my poster under methods, we're looking at orienting behavior. This is where fish are swimming towards each other. They're looking at each other. Following behavior, where they're swimming next to each other in a parallel fashion. And then dispersing behavior, where they're swimming away from each other. And zebrafish at the same time have similar sensory modalities that we do as humans. So they have an olfactory system, they have a visual system, and they also have this mechanosensory system, which is how they kind of sense water pressure and current. So it's not quite the same as our human tactile sense, because the cells along this area are more like our inner ear cells, but it's still an interesting sense that we can kind of compare to tactile sense. And so we can experimentally manipulate these senses. We can take away visual input by just recording the social behavior in the dark. We can affect the mechanosensory input by ablating or killing the cells that allow for mechanosensory information to pass. And then to answer the second part of our research question of which regions of the brain are important for social behavior, we can use this protein called phosphoarc, which is found in many neurons in the brain, to mark neurons that are active. So we can achieve here what looks to be the average average forebrain of a zebrafish when they are alone, and then also the average intensity and signal of a zebrafish brain when they're in a social condition. So you can already see that there's more activity in the forebrain when fish are in a social condition, which is what we would expect, right? Because you're being social, you're doing more behavior that will involve more parts of the brain and activate more neurons. And from these, we can also identify, which region of the brain is social specific, which region of the brain is active only in the social condition. And from here, it's this sort of anterior part of the telencephalon here. So that's all good information to have when we're looking at our data. So from our behavioral results, we can see that there is a significant effect of vision. When vision is ablated, we can see less orienting behavior, less following behavior, and more dispersing behavior. So orienting and following being the most social of the behaviors, those are decreased when we do not have visual input. And at the same time, we don't see any significant changes when we ablate mechanosensation. So we can conclude that vision is the primary driver for social behavior, and mechanosensation isn't all that important. And that was kind of what we expected, although I possibly expected to see an effect with following behavior, because they're tracking close to each other's bodies, and perhaps, water pressure or current or something like that would help maintain that behavior. Now, when we look at brain activity, we see what we would expect. The social control fish, those fish that have vision intact and have their mechanosensation intact and are in a social condition, they have greater brain activity in their forebrains than fish that are by themselves and fish that are in the dark. So again, it's pointing to the importance of the visual system in the brain activity. And we can also support that by looking at, again, this social only region. If we look at the intensity there, we can see indeed that fish that are in a social condition have greater brain activity in the social only region. So that confirms that, yes, this is the region that is important for social behavior. Which again, if you do look at the data, it's not to say that there aren't neurons in that region that aren't active when fish are alone. So we need to gain more information about the diversity of that population of cells in that region. But we can conclude at this point that our social behavior in these zebrafish, at least in the 3 behaviors that we measured, is visually driven. So when we have our vision intact, we have social behavior. When we lose our vision, that social behavior is disrupted or decreases. And we can also see that mirrored in the brain activity, where subjects that are in the social condition and they're visual, they have greater forebrain activity than fish that are by themselves or fish that are in the dark. And again, just looking at our region that's important in the brain, this region includes regions of the human brain. It's been evolutionarily conserved in zebrafish and in humans. And so it kind of points to the importance of learning and memory and those parts of the brain that may be important here. So obviously for future, I want to continue with this experiment and work on olfaction now and understand, what's the impact of olfaction. If we were to, ablate every sensory system, will we just see nothing happening, or how would that affect behavior? And I also want to explore more complex computational methods for understanding behavior. So right now, the way I've been doing it is just, setting parameters and binning, every data point into the 3 types of behavior. But I would love for a computer program to do it in an unbiased way, so we could see more diversity of behavior and possibly, social behaviors that, we weren't able to characterize by observation. And then, of course, analyzing the brain in the 3 dimensions will help us understand anatomical regions that are important, rather than just location-wise. Are there actually evolutionarily conserved brain regions important for social behavior? But that's kind of the rundown of my project. I'm happy to answer any questions you guys have.
Well, folks are typing some of the questions to this wonderful research. Thank you so much for presenting it and so compellingly. Tell me about something that you want to learn in the near future that you think will help with continuing this research.
Right. So what of my kind of 3 future directions, the most easily achievable one is looking at the brain in 3 dimensions. So we have wonderful technology at the University of Oregon, this Amaris technology that allows you to visualize the brain in 3 dimensions, and you can draw 3-dimensional regions of interest. And this was something that I was working on prior to everything being closed down. So I'm hoping that, once I'm able to get back in the computer lab, I'll be able to get that kind of data. But otherwise, I'm looking forward to also writing the programs to do the clustering and the unbiased behavior characterization.
Do we have a question in the chat? Ian, if you're trying to speak, just unmute yourself.
Oh, Okay, question says, is social behavior being visually driven different between species? So that is a good question. And it's definitely something that we seem to think is true in humans. If you think about autism spectrum disorder, one of the characteristic behaviors is lack of eye contact and lack of attending to information. Like if you point at something and say, look over here, they don't look. So it does appear that, you know, vision is important, but there's also another theory in the literature that something is disrupted in the autism brain that has to do with multisensory processing. So, you know, the way that we modulate background noise and kind of filter out which stimulus is important and which is not, that seems to be impaired in people with autism. So that could be, again, something that I want to continue to study, sensory interaction. We didn't really see any interaction in my study looking at these three behaviors, but that's not to say that there isn't any. And so I'm hoping that perhaps olfaction will reveal that, or, you know, using more unbiased ways of looking at behavior will reveal some interactions.
Thank you so much, Adeline.
Another question from Jessica is, what first got you interested in your research question, and how do you foresee any real world impacts down the road? So that is a good question. I actually started doing research in a psychology lab. Abby and I were in the same lab freshman year, I think, which was really great. But I think for me, why I went towards the neuroscience side after working in psychology is I really wanted to, you know, experimentally answer the questions that we're interested in. And, you know, with working with animal models, you have a lot more control over different factors. So you can really get down to the exact mechanism and exact cellular level of what's going on. And so that's something that really interested me. But I did want to, you know, conserve my interest in behavior and interest in psychology. And so that's kind of why this project was perfect for me. And then, do I foresee any real world impacts? Yeah, ideally, if I had, you know, plenty of time and infinite amount of money, I would love to see this project through and hopefully come to, you know, a conclusion about maybe, you know, one sense or two senses that are important. And then that would be inspiration for some occupational therapy options for people with autism. Because right now, I think we do have lots of good evidence that early intervention is really helpful for people to, for autistic people to, you know, gain social behavior competency. But at the same time, you know, it's a spectrum. And so we'd love to make that easier for everybody. And so that's kind of where I hope the research goes. And yeah. Obviously, it's always a team effort. And so I know that my research is only one part of that solution. I think everyone here acknowledges that as well, probably.
Those were such great questions. I was wondering if Ian would be willing to answer them as well. What got you interested in this, and what are the impacts, the real world impacts you see down the road for applications?
So what got me interested in it? So I joined the DJJ lab as a freshman, mostly just to be interested in getting into a lab. I just wanted to do some sort of research, get involved. And through that, I mean, sort of general sort of environmental safety and environmental health is really important, I feel. And our water streams and water health is really important, I feel. And this is kind of just like one sort of subject with sort of just water purification, sort of keeping their water systems clean and healthy is really important. And I feel like that's something I can help through this research. In terms of sort of real world impacts, ideally with adapting my sensors to study not just aqueous solutions, but then also soil samples. Ideally, my FETs or my sensors could be used in sort of like a chemical test kit, and that it could be like used in like on site with the farmer, just like, okay, let's see what's the nitrogen or what's the ammonium concentration in the soil right now. Am I putting too much fertilizer in? Can I afford to put a little more, et cetera, et cetera.
Thank you so much, Ian. Did anyone else have, go ahead.
One question I kind of wanted to ask everybody, those who are working with animal models, is why did you decide to use the animal model that you did? Because I think we're really lucky here at the university to have so many options, whether it's using, clinical subjects and humans to using zebrafish is what I do. So maybe Byron, I know you were using mice. Why was that a good choice for you?
Well, we use mice because that's our Alzheimer's disease model. Like I would say we could potentially create an Alzheimer's disease model in mice that is similar to what we see in humans. So that's why we use mice, because we could potentially it could be representative of what we see in humans.
Yeah, and I would say for our lab, I mean, our research centers around the primate. So ultimately, the research that we do revolving primate behavior helps kind of two facets of the primate world. The first one is looking at conservation and seeing the impact environment has on social behavior, particularly because the environment of the non-human primate all around the world is constantly changing, and we see their behavior evolving and changing along with it. And so it's really important to see and to understand exactly how those behaviors are changing, so we can alter our conservation efforts accordingly. And then the other side of that is non-human primates are also often used for biomedical research, which is what our study group of Japanese macaques are used for at the ONPRC. And again, it's particularly important that their environment and their welfare is taken into consideration when institutions like that are building their enclosures and deciding, how many resources and how to allocate to them, how to provision them, and then also, the size of their enclosure, how big their group should be. And then also, looking at primates at zoos and at rescue centers. So there are a lot of really important reasons to look at primate behavioral research.
I would say that the Labuda lab utilizes C. elegans, and it's particularly really impactful for my work because of a variety of different reasons. So C. elegans is a really powerful model organism because of its high fecundity and fast reproduction time. And also C. elegans exists in two forms. So there's the male form as well as the hermaphrodite sexual form. And so it's very easy. They're also very easy to genetically alter. And then it's very easy to maintain homozygous populations just by maintaining hermaphrodites. And the really cool thing about C. elegans for studying meiosis is that the germline is spatially and temporally organized in this pipeline of meiosis. So we can actually distinguish what stage of meiosis cells are at just by looking at the different chromosome morphologies and following them through the progression of the germline.
Everyone, I was just frozen, so it looks like everyone is moving again. It's a little like I'm having the reanimation spell at our disposal or at the whim of Zoom. Thank you so much. Thanks, Dylan, for the head bobble. Did anyone was Andrew able to get on to there was one other question for Abby that was earlier. Nope. Okay, let me get that for you all. He was also saying that he was frozen, so he might be coming back to join us. Abby, the question for you: did the results that you got give you any insights on helping children with language development?
Great question. That is something that we are really looking into right now with these models that we're building. So my project mainly is kind of fueling this recent research on these theoretical models of language development with children, specifically with bilingual children, because there's so much literature on monolingual children right now, because they're easier to test because you only have to test and look at one language. So with bilingual children, you don't really have to account for both of their languages. So that is one thing that this project is informing, is these theoretical models. And then eventually and hopefully this work can inform clinicians in the field of speech language pathology and serve children who have language delay or other developmental disabilities, and kind of figure out how to best target how they're organizing these vocabulary systems and how those can be targeted in therapy.
Everyone, it is 7:18, and thank you so much for your amazing work, the beauty and the eloquence of the way you presented it today, and for staying with us for a few extra minutes so that we could all ask our questions. We have one person, Dr. Walker, is saying, nice work, presenters, and thank you to the moderators. Oh my gosh, it was such a pleasure to be in the presence of such great, thoughtful, exciting information, and to hear about the work that you've been doing throughout this time. I'm an old person, so I'll say something corny like, there's a lot to worry about, but I'm really reassured knowing that we have the kind of work going on that that you all were able to talk about so eloquently today. Going forward, I think we're in really good hands. And I'm really excited to actually see if we can get some sort of flaming banana bread without rust, however, Dylan. So thank you all so much for everything. Andrew, I don't know if you've been able to make it back. I don't know if you have any final words. I'll give him a moment.
Hi, I'm here. I just thank you all. This is so far out of my area of expertise, and you've all blown my brain out of the water. So congratulations and thank you.
We have amazing people here at the University of Oregon, especially our undergraduates. Thank you so much. High five. Woo, 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 woo. All right. See you all later. Thank you. Can't wait for next year to hear what you've done next.
Thank you.
Bye.
Thank you.
Thank you.
Congratulations, everyone.
Thank you.
Bye all. Thank you so much.
Thanks,
Bye.
Jessica.
Thank you.
I tried to follow along to just see what it looked like on the YouTube live, but my computer is super flaky, so I was able to pull it up